Can everyone see my screen?
Yes.
Okay, good. So this is Dr. Garais, April 6th, week 4, 6 p.m. And this is MED 110 is this class, 5 NAX. So it's Tuesday evenings. And what is due this evening for week 4, of course, task 4, not task 4, task 3, discussion 3, and lesson 3 is due tonight at 6 p.m. But you guys know me, if you can get it in by like 10:30 tonight, I may look the other way on taking points off. So please, I see 3 people, Ms. Kamara, Ms. Desiree, Ms. Jones. That's what we're starting with. Let's hope others come in soon. Ms. McGuire is also here. Lovely. And Ms. Ciara as well. And whoever is coming in on iPhone, kindly in the chat, put your full name so I know it's you. So what are we doing this evening? Well, we were continuing on, and we already talked about this. It's our theme of Anatomy and Physiology 1. All right, Ms. Salvatierra, I got you. And again, this is the theme of the last 3 weeks and including this week, which is this picture right here. So I'll make this a little bit bigger. Atoms, bonds, molecules, and we talked about the organelles and the cells, and we talked about tissues. Now we're going to talk about how all of that gets directed. And we already had a little introduction of it last week when we talked about DNA, okay, deoxyribonucleic acid. And as a quick review, it was in the, say it to yourself at home, it starts with an N, your nucleus. And remember, there was chromatin in the nucleus, and the chromatin is the loosened up DNA coils, and that gives us the ability to now look at the genetic code. And if you look at what we're going to do this week, we're going to talk about the nucleus, DNA replication, protein synthesis, cell growth and division. So what is the structure and function of your DNA? And if you look at lesson 4, there's this lovely video that we're going to break down right now. I mean, you could look at a textbook and notes, but this video right here, it's got it all. It's and we're going to take it step by step. So don't worry about all this stuff, but this video is golden because it's going to be talking about all the things that we, a lot of our goals for this week.
It's just beautiful, isn't it? It's mesmerizing. It's double helix sighting. You really can tell just by looking at it how sort of important and amazing it is. It's pretty much the most complicated molecule that exists and potentially the most important one. It's so complex that we didn't even know for sure what it looked like until about 60 years ago. And so multifariously awesome that if you took all of it from just one of our cells and untangled it, it would be taller than me. Now consider that there are probably 50 trillion cells in my body right now. Laid end to end, the DNA in those cells would stretch to the sun not once, but 600 times. Mind blown yet? Hey, you want to make one?
Okay, that's the theme of this whole video is that, and you're going to see he's going to keep on talking about how much information gets packed into such a little tiny thing. And remember, the cell is only so big. We're talking about the nucleus, the thing that's inside the cell. Remember, it was the brain, it houses the DNA. And we also talked about in week 1 and week 2, excuse me, where the hydrogen bonds of the DNA give the DNA its ability to compress and to supercoil so that everything can fit in such a tiny little space. And that should freak you out a little bit. In that tiny little space, it has everything of who you are, how tall you're going to be, what color your eyes, are you going to have long, not long, but are you going to have thick hair or thin hair? And also now, according to the American Psychiatric Association, 40% of your genetic code has behavior. So that's a big, big thing now regarding mental illness. That's a big thing now also regarding alcoholism and also drug abuse, because there was always a debate, nature versus nurture, and DNA is the nature of who you are. Okay, doesn't mean that, oh, if you have genetics to be a killer, you're going to be a killer or a psychopath or whatever, right? But there is a greater potential of it. And that is the nurture part, what you're exposed to. So just know that there's two sides to human beings, but we're going to be talking about the nature side, the programmable side, which is your genetics. And it has, just think about everything that you are, every cell. It has, the DNA has the instructions for everything. So now all that supercoiling, hydrogen bonds, now that all makes more sense. And remember, your body was designed. It's not made up, you know, it's not random. There are specific things. And that's what DNA is. It is the blueprint of who you are. And now there's scientific evidence that states that even your behaviors are also potentially inherited.
Of course, you know, I'm talking about deoxyribonucleic acid, known to its friends as DNA. DNA is what stores our genetic instructions, the information that programs all of our cells' activities. It's a 6 billion letter code that provides the assembly instructions for everything that you are. And it does the same thing for pretty much every other living thing. I'm gonna go out on a limb here and assume that you are a human, in which case every body cell that you have, or somatic cell in you, has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed together tightly with proteins in the nucleus of the cell. DNA is nucleic acid, and so is its cousin, which we'll also be talking about, ribonucleic acid, or RNA. Now, if you can make your mind do this, remember all the way back to episode 3, where we talked about all of the important biological molecules, carbohydrates, lipids, and proteins. Now, ring a bell? Well, nucleic acids are the fourth major group of biological molecules, and for my money, they have the most complicated job of all. Structurally, they're polymers, which means that each one is made up of many small repeating molecular units. In DNA?
So think of polymer as like a monomer is like a building block. And poly means many, mono means one. And think of it like Lego. So they all connect together and then it forms this chain.
Small units are called nucleotides. Link them together and you have yourself a polynucleotide. Now, before we actually put these tiny parts together to build a DNA molecule like some microscopic piece of IKEA furniture, let's first take a look at what makes up each nucleotide. We're going to need 3 things. 1, a 5-carbon sugar molecule, 2, a phosphate group, and 3, one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They're like the genetic coding that makes you. 2, a phosphate group. And
so what makes DNA? Doesn't this look like, since we have midterms next week, and I'll talk about that later, doesn't this look like a beautiful all of the above question? So how do I know I have a 5-carbon sugar? You see this is shaped like a pentagram. 1, so there's a carbon here at this point, a carbon here, a carbon here, and a carbon here connected via double bonds to an oxygen. So ribose, which is a 5-carbon sugar, hence the term deoxyribonucleic acid. Now, you've got a phosphate group, and then you have your choices of 4 nitrogenous base. So a monomer, right, can be made up of, you definitely have a 5-carbon sugar, ribose, then a phosphate group, and then one of these, adenine, guanine, cytosine, and thymine. And then you could see how these things, can form different combinations. And let's show that now.
Be one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They're like an intertwined microscopic double spiral staircase, basically just a ladder, but twisted, the famous double helix. And like any good structure, we have to have a main support. In DNA, the sugars and phosphates bond together to form twin backbones. These sugar phosphates.
So you got to think of DNA like a ladder. So there's 2 of them. So you have your phosphate group, and then you have your deoxyribose. These 2 together form, like, the rungs of not the rungs, but the backbone of the ladder. And then the bases, either adenine, thymine, guanine, or cytosine, will now form the rungs or the steps of this ladder.
Bonds run down each side of the helix, but chemically in opposite directions. In other words, if you look at each of the sugar phosphate backbones, you'll see that one appears to be upside down in relation to the other. One strand begins at the top of the first phosphate, connected to the sugar molecule's 5th carbon, and then ending where the next phosphate would go, with a free end at the sugar's 3rd carbon. This creates a pattern called 5' and 3'.
Now, just know that, if you haven't had your biochemistry yet, just know that 5' and 3' is just simply a direction. And if you notice here, you see the oxygens are pointing up. So this part of the ladder is going to move in this direction, facing down. And this part of the ladder is going to move up. So they're going to be, so this side is called the 5' to 3', heading that direction. And this is heading the exact opposite direction. So this one will start at 5' here and then move up, and then 3' will be up here. So that's one direction. And then the other one is going in, the other side is going in an exact opposite direction. And then also notice here, if you look at your nitrogenous bases, A will always match to T, and C will always match to G. So if there's a T here, it'll match with A. If the G here matches with C. If the C is here, it will match with G. If an A is here, it'll match with T.
The deoxyribose with an arrow, with the oxygen as a point. It always points from 3' to 5'. Now on the other strand, it's exactly the opposite. It begins up top with a free end at the sugar's 3rd carbon, and then the phosphates connect to the sugar's 5th carbons all the way down. And it ends at the bottom with a phosphate, and you've probably figured this out already, but this is called the 3' to 5' direction. Now it is time to make ourselves one of these famous double helices. These 2 long chains are linked together by the nitrogenous bases via relatively weak hydrogen bonds. But they can't be just any pair of nitrogenous bases. Thankfully, when it comes to figuring out what part goes where, all you have to do is remember that if one nucleotide has an adenine base, only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you. So AGGTCCATG means something completely different as a base sequence than, say, TTCAGTCG. Human chromosome 1, the largest of all of our chromosomes, contains a single molecule of DNA with 247 million base pairs. If you printed all of the letters of chromosome 1 into a book, it would be about 200,000 pages long. And Each of your somatic cells has 46 DNA molecules tightly packed into its nucleus. That's one for each of your chromosomes. Put all 46 molecules together, and we're talking about roughly 6 billion base pairs in every cell. This is the long.
Now, you could see where this could lead into problems. If the code is messed up in any way, don't you think it's going to shift all the letters over? It's just like in an exam. You know, if you mess like next week you're going to have a 50-item multiple choice examination. Just imagine you skipped one and then it shifted all your answers over. Well, that's one of the problems with DNA, is that there could be errors. And we now know that in genetic disease, if the DNA is wrong, then the code will be wrong. And if the code will be wrong, all the other things that we talked about last week and the week before, you know, the cells, the organs, and the tissues, all those will be wrong as well. So you could see how the importance of genetics and the importance of the study of these base pairs and the code is really important. And it's also highly complex. It's not an easy thing. It took 30 of our best scientists several decades just to even map out the code. And that was in the early 90s. And one of my mentors in biochemistry, she was part of it. And it's just very, very complex. So let's look at, I want to skip to when we're talking about how to write out the code.
Short strand of DNA. I'll give you one base sequence. You give me the base sequence that appears on the other strand. Okay, here goes. So we got a 5' AGGTC.
Okay, so here is a classic question that's on, that could be on my exam or anybody else's exam. So let's take a moment to answer that question. So, ooh, what's this? Let's look at the code that he just gave. Now, this particular code started at 5', then it went A, G, G, T, C, C, G. And this a little bit closer. And of course, if you start at the 5', you're going to end at the 3' here. Now, a classic question would be, now we definitely know that this is DNA, right? You got 5', 3', and they have adenine, guanine, thymine, and cytosine. So that already, I know DNA, and DNA has 2 strands that are connected to each other. Okay, so the way to answer this is you put down 3' on one side, and you know 5' will end up on the other side. And then you just match up the pairs. A will always match with a T. A G will always match with a C. A C will G again will always match with a C. A T will always match with an A. C will always match with a G. G, and a G will always match with a C. And if I started at 3', I'm going to end at 5'. So this could be the question. If given one strand of DNA below, what is its complement? Below or complement strand. That's a classic question. And of course, this would be your answer. And the way to get this is, of course, you always start, because these two things are going the opposite direction. This one's going 5' to 3', and this one, the other part of the DNA, will always go from 3' to 5'. And they'll connect. And the connection in between these base pairs is hydrogen bonds. And that's why the staircase kind of twists and can be compressed and compacted. So let's see if we're right. TCCAG, what was it? TCCAGGC. So let's see if we're right.
G to 3' and time's up. The answer is 3' TCCAGG.
So let's see if we're right. TCCAGGC and going from the 3' to the 5'. So pause the video, try it on your own, and see how I got it when you go and study later, because I can guarantee you there's going to be a couple questions like this, and also for future NCLEX, they could ask you something similar to this. All right.
5'. See how that works? It's not super complicated. Since each nitrogenous base only has one counterpart, you can use one base sequence to predict what its matching sequence is going to look like. So, could I make the same base sequence with a strand of that other nucleic acid, RNA? No, you could not. RNA is certainly similar to its cousin DNA. It has a sugar.
Okay. Now, ribonucleic acid is different. Of course, it has the phosphate chain, and it has ribose 5 sugar, but right has this extra oxygen. That's why DNA is called DNA, deoxyribonucleic acid, and RNA is called ribonucleic acid. Now, DNA, if DNA is the blueprint of who you are, RNA is kind of like a foreman or a messenger that brings the DNA code out of the nucleus and into the hands of the ribosome. And if you recall last week, the function of the ribosome was to make proteins. And the only way you could do that is if you had the proper code. Now, RNA is only single-stranded, if you look here, right? And what else do we see that's different? It has an adenine, has a cytosine, has guanine, but instead of thymine, it has uracil. So that's how you know you have RNA. It's only single-stranded, there's an extra oxygen over here, and there's a uracil nitrogenous base hanging around.
A backbone with nucleotide bases attached to it, but there are 3 major differences. 1, RNA is a single-stranded molecule, no double helix here. 2, the sugar in RNA is ribose, which has 1 more oxygen atom than deoxyribose, hence the whole starting with an R instead of a D thing. And finally, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. RNA is super important to the production of our proteins, and you'll see later that it has a crucial role in the replication of DNA. But first.
Okay, now we're going to skip that part. Now let's go back to, because that's the history part and your discussion is about the history. Now let's go back to our DNA here. So this, if you put this all together like this is one, if I'm looking at this, this is a DNA, a strand of DNA. How do I know? Here's one string and here's another string. One is going from 5' to 3', the other one is going 3' to 5'. And there's adenine, guanine, cytosine, and thymine. That's how I know I have DNA. Now, how do I know I have RNA? Well, how about if I wrote it like this? And this alone, right, is only a single strand. And instead of thymine, it has a uracil. Do you see how this strand can match up with this strand up here? And I'll color this red. So we're going to talk about how we're going to unzip and we're going to separate these 2 strands, and the RNA is going to copy itself, because remember, I need the DNA code, right? And let's insert Nice shape, down arrow maybe. Okay. I have my DNA code, and then, ugh, that's just giving me trouble. Put this over here. I have my DNA code, then RNA is going to copy a little bit of it. Make this arrow a little bit smaller. Copy this arrow. So this is what has to happen, right? And I'm going to draw a dotted line here. This all happens inside the nucleus. This is outside the nucleus, either in the cytoplasm or the rough endoplasmic reticulum, which was the factory where the ribosome will now make proteins. So the ribosome now will make proteins, and the proteins have to be amino acids connected by peptide bonds. And they have to be, the amino acids have to be in a specific order. Hence, you could see the code runs in a specific order. So you have to be amazed by this, that the DNA code has to get copied correctly into the RNA code. Then it goes out of the nucleus, out into either the cytoplasm or the rough endoplasmic reticulum, where it hits these ribosomes. The ribosomes then put together amino acids with a peptide bond, and its product is to make proteins. And the proteins get to do things. And we'll learn in anatomy physiology too, like hormones and neurotransmitters. Proteins have a lot to do with what direction does the cell go in? What's the cell going to go do? And what's the cell's function? So you could see here, if I have a messed up DNA code, it's going to mess up my RNA. It's going to mess up the ribosomes. The ribosomes won't be able to make proteins, and then the amino acid sequence will be off. A classic example is Down syndrome, right? You have non-disjunction of some of your genetics. That means it doesn't break apart, and the code will get wrong. And then the RNA will copy it wrong. The ribosomes will then get the wrong instructions, and then the wrong proteins get made. And then what happens? Down syndrome. Okay, the low set ears, mental retardation, early onset Alzheimer's, and of also what do you call that? The they have a classic predisposition of fat, right, on their body. So this is all from what? From the DNA code being copied onto the RNA code. And I could ask you, where's the DNA? Well, it's in the nucleus. And remember, we talked about last week, when it's all compact, it's in the nucleolus. And then when it gets relaxed, it's in chromatin. And then that's where the RNA then can copy itself and then go out of the nucleus and then go to the ribosomes and make some proteins. So know this and know this pathway, because it's pretty much is what happened to you every second of your life. And the other important thing is, now it's apropos because we're talking about viruses, right? Like HIV. Okay. So what does HIV do? It's a retrovirus, right? It hijacks the DNA. And then what does it do? Go back up here, tells the DNA, hey, we're not making what you want to make anymore. We're going to make HIV proteins now. Okay. So that's also how coronavirus works, right? It'll go right through the cell wall, right through everything, all the way up, hijack the DNA code, and then tell the DNA to do something that it didn't originally want to do. And remember, what does a virus want to do? It just wants to live. It's not a good thing nor a bad thing. So now we already know the structure of the code. And now we're going to talk about a little bit, and now we know how to read the code a little bit, at least how to match up base pairs. Now let's talk about how to actually copy the code. And I'm going to skip a little bit ahead because they're going to start talking about Watson and Crick and that's more for your discussion. And it's very interesting, like how all the stuff that you learned in your history books, they're just like, it's just like any fake news or anything, because you think about it, who writes the history books? The victors, the people who did win. But remember, there's other people's perspective. And it's only recently, for example, we always thought that Columbus Day, it's a real Italian holiday. No, it isn't. And then when you really read exactly what Columbus and his little buddies did, it's not the bright and shiny history that people like. And the same thing in science as well. History gets rewritten. The Big Bang Theory now is now being contested because now recently we found some errors in the math. So the thing that we thought was real, and that's the good thing about science. Someone's always looking to see if your numbers are right. And that's why we always tell you guys, find vetted sites, find the most updated site, because even if you get something from a .gov.edu from 10 years ago, it could totally change. A classic example, a couple years ago, I went to a diabetics update conference, and they gave like a mini quiz. And I'm like, oh, I'm in internal medicine. I should ace this quiz. I failed it because the stuff that I learned about diabetes was like 10 years ago. And there's way more things that come out that we manage diabetes much better now than we did 10, 15 years ago. So always try to find not only vetted sites, but something recent. So if you get something from like 2015, 2016, I'm always going to have a comment like, hey, wasn't there something you could have found like closer to 2019, 2020, and get into that habit.
Extremely good at this. Our cells can create the equivalent of 10.
Yeah.
It started from an original copy and will copy itself trillions of times over the.
So how does the DNA copy itself onto the RNA so that the RNA can give that code to the ribosomes and to make us some proteins?
Lifetime. Each time using half of the original DNA strand as a template to build a new molecule. So, how is a teenage boy like the enzyme helicase? They both want to unzip your genes. Helicase is marvelous. Unwinding the double helix.
Bad. All right. And also potentially inappropriate. Now, enzymes. We already know what enzymes do. We learned that week 1. Enzymes break down things, and they speed up processes. Now, helicase, the double helix is the DNA. Now, when we look at this, I have one strand going this way, another strand going this way, and I have ATGC on both sides. Does everyone see that? So that's how I know it's DNA. So you have an enzyme called helicase. ASE is the suffix that means enzyme. So it's going to break down this double helix. So we're going to have to break down the DNA and unzipper it. See how it forms a zipper? And it goes so that it can now expose the two strands. And this strand, well, I'm jumping ahead of ourselves, okay? So I could ask you, what enzyme unzips DNA? Helicase, okay? So that's one potential question.
Next, speed slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand, as I call it. Another bottom strand called the lagging strand, which I like to call the scumbag strand, because it is a pain in the butt to deal with. These unwound sections can now be used as templates.
You're going to see what he means by that. Now, leading strand just means that if you look at it, it goes from 5' to 3', and the other enzymes love going in that direction, 5' to 3'. The problem is the other side is 3' to 5'. So some of the enzymes are going to have to work backwards. And you'll see what we're talking about in a minute. And that's why they call it the lagging strand. And if you have to work like backwards or sideways, it's going to take you much longer than it would if you were the leading strand. But still, again, this is all designed. We're not making this stuff up.
Create two complementary DNA strands, but remember the two strands go in opposite directions in terms of their chemical structure, which means that making a new DNA strand for the leading strand is going to be much, much easier than for the lagging strand. For the leading good guy strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down molecule. But before it could do that, it needs a selection of nucleotides that fill in the section that's just been unzipped. To get started at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook onto so that it can start building the new DNA chain. And for that little primer, you can thank the enzyme RNA primate.
So you need a primer. And what's prime? Prime means first. So you need RNA primase to start the thing, right? And how do I know it's RNA? Look at this, AG, and that's not a T, that's a U. So that's how I know that this is a matching strand to this unzipped DNA, that this is now RNA. And RNA primase facilitates that. Then the building of the rest of the copy can be made by DNA poly, also known as DNA polymerase. And poly means many.
So the leading strand only needs this RNA primer once at the very beginning. Then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. Copying the lagging or scumbag strand is, well, it's a freaking scumbag. This is because DNA polymerase can only copy strands in the 5', 3' direction, and the lagging strand is 3', 5'. So DNA polymerase can only add new nucleotides to the 3' end of a primer. So maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments. And here that awesome little enzyme, RNA primase, does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. These are called Okazaki fragments.
So you see that word Okazaki fragments? That means you're dealing with the 3' to 5' strand, and that means you're walking backwards. So you do like this part, and then you go that way. Then do this part, and then you go that backwards. Then this part, and then backwards. So that's why they call it the lagging strand, because it takes longer. So, and they go in these fragments, and these fragments are called Okazaki fragments. But it gets formed the same exact way. You need a RNA primase to start it, and then a DNA polymerase, then to finish out the rest of the chain.
Couple of married scientists who discovered this step in the process in the 19th.
Oh, by the way, doesn't that look like a beautiful question? Which of the following complete the chain? DNA polymerase, DNA primase, RNA primase, or what was the first one? DNA helicase. Which of the following break down the DNA into two separate strands? DNA helicase, DNA primase, Okazaki fragments. You could easily pretty much figure out my exam, right? By just mix and matching. So what am I saying is that these enzymes, they're important, and you will see it.
And thank goodness they were married so that we could just call them Okazaki fragments instead of Okazaki someone fragments. These allow the strands to be synthesized in short bursts, and then another kind of DNA polymerase has to go back over and replace all of those RNA primers. And
yeah, they don't want to confuse things. There's DNA poly 1 and poly 2. But just know Okazaki fragments, it's 3' to 5', also known as the lagging strand.
then the little fragments gets joined up by a final enzyme called DNA ligase. And that is why I say that the lagging strand is such a scumbag.
Now, what does the word ligand mean? Means like to glue everything together. So that's the last one, DNA ligase. And you could see how the process on the lagging strand is much more complicated, but just as important. Now, you can also see now that it makes an exact copy. And that's what we want when we have either replication of the cell, like in a somatic cell, soma, your body, like, you want to grow more skin cells, right? So the DNA needs to split, and then the cell needs to look and act just like its parent cell.
DNA replication gets it wrong about 1 in every 10 billion nucleotides. Don't think your body doesn't have an app for that. It turns out that DNA polymerases can also proofread, in a sense, removing nucleotides from the end of a strand whenever they discover a mismatched base. Because the last thing we want is an A when it would have been a G. Considering how tightly packed DNA is into each one of our cells, it's honestly amazing that more mistakes don't happen. Remember, we're talking about millions of miles worth of this stuff inside us. And this, my friends, is why scientists are not exaggerating when they call DNA the most celebrated molecule of all time. So you might as well look this episode over a couple.
So DNA poly, you can see it has two functions. One, of course, it builds the rest of the copy of the DNA. And two, it has a proofreading. So every 10 billion pairs or so, they just make sure that A's matches with G's, T matches with C's. Now, this is another thing I wanted to show you. That relates to what we were talking about before. Here you go. Remember the bonds? Remember the hydrogen bonds, right? They're partial, the same thing with water. There's a partial negative that gets attracted to a partial positive. Looky here. You have a partial negative here, partial negative here, partial positive, partial positive. So for all AT and AU, there's only 2 hydrogen bonds. For all GC or CG, there's what? 1, 2, 3 hydrogen bonds. And because of that mathematics, right, that's why you have the staircase starts winding and turning. You don't want to know the math of that. That's in a class called Cell Molecular Biology in graduate school. Please, if you take that class, have it help you. All right, enough reminiscing. So we now know the structure of DNA. We now know the structure of RNA. We know how it copies it. We know how it reads it, and also how it proofreads it and how it controls it. Let's look at stuff in task 4. So for nucleus, what we know now, the DNA is in the nucleus and how it replicates. We now, let's look at protein synthesis, because remember last week I said I didn't like that protein synthesis video that was in lesson 3, and we're going to talk about cell growth and division in a second. So let's look at protein synthesis. Let me find a better video because I don't like that one. Where's the one I like? I think it's this one.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
Do you see how it's like coiled? And then this protein called histones makes it even super coiled. And actually, when we're looking at a chromosome, all it is like all these coils upon coils upon coils of just DNA. And that's how we can fit all of that information in such a small space. And it's because of the hydrogen bonds between adenine and thymine, which there are 2, and guanine and cytosine, which there are 3.
Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus.
Can you see that this is single-stranded? This is double-stranded that just got unzipped, and we already know about helicase that can do that for us.
The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
Now, either in a loose ribosome that's floating around in the cytoplasm, or the ribosome that's connected to the rough endoplasmic reticulum, which is our main protein factory.
Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid.
And these are amino acids, and they're connected by peptide bonds, and you're forming a chain, and that chain is called a protein. You can also see it reads it 3 at a time. Can you just imagine if this one got messed up and then all the letters shifted over one? Then this whole chain would be what? Garbage.
Is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
And that is what's happening every single second of your life. And it's highly complex. You could spend your entire life trying to figure that out. And right now, science, we know what the code looks like, but we're like, Dr. Garais was she was my mentor for anatomy and physiology, and also helped me get into medical school. She said because we were like, it's like an amoeba, amoeba, like it doesn't have a brain. It's like amoeba trying to drive the space shuttle. That's how much we don't know about what just went on. Let's look at what we just shown there in this. So we have DNA code going to RNA. So in that area from here, right? Let me put a text box. We mentioned a special kind of RNA, and that's messenger RNA. So the messenger is going to take that copied code, and they're going to take it, and then they're going to put it outside the nucleus, either in the cytoplasm or the rough ER. And then once it gets to this ribosome right here, which is the protein maker, then what's going to happen? You're going to have a, where's my text box? tRNA. or transfer. So the code will now get transferred onto this ribosome, and then the ribosome will make proteins in the form of those little, purple fuzzy balls. Those are amino acids, and they get connected together with a peptide bond. And this is happening every single second, even before you were born. And it's highly complex. And you could see there's so much room for error, but it's amazing. That's why we in the healthcare field, we cherish life, all life, even if the life is annoying, right? For those of you who got kids. But it's important, and you could see how important it is. And also, another brilliant thing about this is you can now see that life is not random. It is engineered and it's designed. So it should be well protected. So this is this video from DNA to Protein 3D. Nice video. I like it better than that other video, the one that was like orange red and had the black dots and that kind of. So that's protein synthesis. So now you know an mRNA, tRNA, where exactly in the cell. And remember, all of this is going on inside your cell, each and every one of your cells, the billions of cells you have. Now, let's talk about this mitosis versus meiosis, which is cell growth and division.
Before we go in depth on meiosis, I want to do a very high-level overview comparing mitosis to meiosis. So in mitosis, this is all a review if you've watched the
Oh, this is pretty cool because this is medical college aptitude test stuff. MCAT. Ugh, hated MCAT.
This video. In mitosis, we start with a cell. We start with a cell that has a diploid number of chromosomes. I'll just write 2N to show it has the diploid number. For human beings, this would be 46 chromosomes. 46 for 46 for humans. You get 23 chromosomes from your mother, 23 chromosomes from your father, or you could say you have 23 homologous pairs, which leads to 46 chromosomes. Now, after the process of mitosis happens, and you have your cytokinesis and all the rest, you end up with two cells that each have the same genetic information as the original. So you now have two cells that each have the diploid number of chromosomes. So 2N and 2N. And now each of these cells are just like this cell was. It can go through interphase again, and it grows, and it can replicate its DNA.
The important part about differentiating mitosis from meiosis is what do you start with, and what do you end up with? So for mitosis, you start with 2N, meaning a complete cell. 23 plus 23. So 1N is 23. 2N is 46. And what do we mean by 23 and 46? 23 chromosomes, 46 chromosomes to make a complete human being. So this is what happens in somatic cells. So if I start off with a skin cell, what happens in mitosis? I will end up with what? The same exact cell, same exact copy, same amount of genetic material, which is 46 chromosomes. So I start off with 46, I end up with 46 chromosomes. 46 chromosomes, another way to look at it is 2N genetic material.
Centrosomes and grow some more, and then each of these can go through mitosis again. And this is actually how most of the cells in your body grow. This is how you turn from a single cell organism into you, or for the most part, into you. So that is mitosis. And one way to think about it, it's a cycle. After each of these things go through mitosis, they can then go through the entire cell cycle again. And let me write this a little bit neater. Mitosis. That S was a little bit hard to read. Now, what happens in meiosis? What happens in meiosis? I'll do that over here. In meiosis, something slightly different happens. And it happens in two phases. So you will start with a cell that has a diploid number of chromosomes. So you will start with a cell that has a diploid number of chromosomes. And in its interphase, it also replicates its DNA. And then it goes through something called Meiosis One. And in Meiosis One, what you end up with is two cells that now have a haploid number of chromosomes. So you end up with 2 cells. You now have 2 cells that each have a haploid number of chromosomes. So you have N and you have N. So if we're talking about human beings, you have 46 chromosomes here, and now you have 23 chromosomes in this nucleus, and now you have 23 in this nucleus, but you're still not done. Then each of these will then go through a phase, which I'll talk about in a second, which is very similar to mitosis, which will duplicate this entire cell into 2. So actually, let me do it like this. So now this one, you're going to have 4 cells that each have the haploid number of chromosomes. And they all don't necessarily have the same genetic information anymore. Because as we go through this first phase right over here of meiosis, and this first phase where you go from diploid to haploid, right over here, this is called Meiosis One. Meiosis One, you're essentially splitting the homologous pairs. And so this one might get some of the homologous, some of the ones that you originally got from your father, and some of the ones that you originally got from your mother, some of the ones that you originally got from your father, some of the ones that you originally got from your mother. They split randomly, but each homologous pair gets split up. And then in this phase, Meiosis II, so this phase right over here is called Meiosis II. It's very similar to mitosis, except you're now dealing with cells that start off with the haploid number. It's important to realize meiosis is not a cycle. These cells that you have over here, these are gametes. These are sex cells. These are gametes. These can now be used in fertilization. If we're talking about if
you recall your maybe your 10th grade biology, gametes are sex cells. So it either could be sperm or an egg. And that makes sense because this only has 23 chromosomes. So in the male testes, each one of these N, one N or 23 chromosomes, this will form a sperm cell, this will form a sperm cell, this will form a sperm cell, this will form a sperm cell. And in female, then it'll form an ovum or an egg. So this will be an egg, that will be an egg. And that's why you need 1 from your dad, 1 from your mom, and that makes you. So 1 N from your mom, 1 N from your dad makes 2 N, and 2 N is 46 chromosomes. That's you, right? So what could I ask? What do you start off in mitosis? 2 N. What do you start off in meiosis? 2 N. Also known as 46 chromosomes, also known as diploid. What do you end up in mitosis? A somatic cell, an exact copy of the original cell. And mitosis, its function is for somatic cells. But in meiosis, what do you get at the end? You only get 1 N or 23 chromosomes, and you get either sperm or eggs, and those are gametes. Okay. I could also ask you, what happens in Meiosis One? Well, the 2N gets split into a 1N and 1N. Now, the neat thing about Meiosis One is the genetic material, this now becomes random, okay? Because remember I mentioned biodiversity in another lecture? We need, because we can't keep on, you cannot marry your sister and have children with your sister or with your brother. The genetics are too close. The genes have to be spread apart and have to be mixed and matched. If they're all the same genes, then it's going to cause a lot of problems genetically, and eventually with the children. And that's why, kings and queens, they have a lot of blood disorders and all these other things, because why? The decades and decades of the intermarriage between first cousins and that kind of thing, right? That's what the function of meiosis is to do what? Mix and match all this genetics so that then when 1N meets another 1N, it'll make a unique human being. And each human being's DNA, each human being's code is definitively unique. That's why when we say we broke the mold the day you were born, it's not an exaggeration. And for people who are trying to get a match for organ donation, you guys know, you probably know what I'm talking about. It's very, very hard to find even a close match, even within family members. So we now know the structure and function of DNA. We know how DNA replicates. We know the difference between mitosis and meiosis, what we start with, what we end with, and that's called cellular differentiation and cell growth and division. And we know about protein synthesis, and we know about the DNA. We know about how the DNA replicates itself and its actual structure. So let's see if what's due next week. So of course, what's due next week. Now, I'm going to cut you a break. No lesson 4. I repeat, do not do lesson 4 because you should be studying for your midterm exam. The only thing to do is task 4, and what's discussion 4 is read about, and that's the part that I skipped in the video about the Watson and Crick. They discovered DNA, but there was this woman named Franklin, right? Ms. Franklin, I forgot her first name. They kind of plagiarized off of her and researched that, and just do quick short answers. And of course, with where did you get the information on? Do you believe Watson and Crick should have gotten the Nobel Prize for chemistry and biology, or should it have gone to Ms. Franklin? And again, back to what we were talking about before, things over history and time. There was once upon a time people thought the world was flat. Well, unfortunately, we still have people who still think the world is flat. There was also a time that we thought demons, demons and the blood in your demons caused illnesses. It wasn't too long ago. So look at that, this discussion 4 with those kind of eyes, with like skeptical eyes about, what's the truth about your history, about everyone's history. So only task 4 and discussion 4 are due for next week. And why? Because we will have a midterm exam, 50 items, multiple choice, based on anything that we went over week 1, week 2, week 3, week 4. And it is cumulative. And I will be putting instructions on about the exam in your announcements. And I will also be putting a practice exam later on this evening so that you can have something to practice during this week, and also get to kind of know, hey, what kind of questions could come out. So on that note, I'm going to stop the recording.